So this is a quick overview of transcription and translation. First, you need to understand the difference between RNA versus DNA structure. So first, RNA contains a ribose sugar. DNA contains a deoxyribose sugar. The deoxyribose sugar has a hydrogen at the 2' carbon, whereas the ribose sugar has a hydroxyl group at the 2' carbon. Furthermore, RNA does not contain the nitrogenous base thymine. Instead, adenine binds with uracil. Uracil, like thymine though, is a pyrimidine, and remember, a purine always binds with a pyrimidine. Okay, so first we need to transcribe DNA into what we call messenger RNA or mRNA. This is where people always get confused. DNA replication does not occur prior to transcription, or it's not needed for the DNA to be transcribed. So because you're making an RNA strand, remember that the RNA is not going to have thymine. You're only going to have uracil. The DNA template is again read 3 to 5, and the mRNA is built 5 to 3. This all occurs in the nucleus. Remember, DNA is polar. It can't go through the nuclear envelope, so all this is going to occur in the nucleus. Certain things have to happen to the mRNA strand in order for it to be able to leave the nuclear envelope. So after transcription occurs, you need what are called post-transcriptional modifications. So the first mRNA strand we make is called pre-mRNA because it hasn't been modified yet. 3 modifications are going to take place. There's going to be RNA splicing, capping, and polyadenylation. So first, what we're going to do is we're going to cut out the obsolete base sequences, which we call introns, and we're going to keep the sequences that will be expressed. These are called exons. So when we're basically cutting out the introns, this is what we call RNA splicing. And it's done with the help of small ribonuclear proteins, or SNRPs, that form a spliceosome with other proteins. And so the spliceosome is going to take part in this RNA splicing to remove the introns, or obsolete sequences. Next, we're going to have capping, and capping is just the addition of a guanine sequence to the 5' end of the mRNA molecule. That is why we call it the 5' cap. It protects the RNA from what are called ribonucleases, which are attracted to phosphodiester bonds, and we don't want them breaking those phosphodiester bonds of the backbone, right? There's also polyadenylation. This is the addition of 250 adenines to the 3' end of the mRNA. So we call it a poly-A tail. This protects the 3' end from the ribonuclease digestion. Also together, when the RNA is leaving the nucleus, it's going to protect the RNA from degradation. All right, translation occurs in the cytoplasm where the ribosomes are located. Right? So you can have free ribosomes, or you can have ribosomes that are attached to the ER. Some of the key players in translation are the messenger RNA. The ribosomes, sometimes you'll see them called ribosomal RNA, and then the transfer RNA. So what happens is that mRNA leaves the nucleus and it attaches to a ribosome. Depending on what type of amino acid you want to make or what type of protein you want to make is going to dictate whether the ribosome is free or attached. The base sequences of the mRNA are translated in groups of 3. These are called codons. Once the mRNA and the ribosome are together, the transfer RNA is going to begin bringing associated amino acids. You'll notice on the ribosome there is an A, P, and E site. A stands for arrival. This is where the transfer RNA is going to arrive. It's going to arrive in the A slot. P stands for polypeptide chain, so that's where the polypeptide chain is going to grow, and E is the exit site where the transfer RNA is going to exit. Because you have to think of the mRNA as consistently moving, right? Moving through. So once the mRNA are together, those transfer RNA are going to bring associated amino acids. The transfer RNA carry what are called anti-codons. And these anti-codons bind to the codons and translate those codons into amino acids to form that polypeptide chain. Now, when you're translating messenger RNA, you are translating the codon, not the anticodon. And since codons consist of 3 bases, you'll see this amino chart helps you translate based on the position of a base within a codon. You have the first letter, the second letter, and the third letter. Translation generally begins at a start codon, or a methionine, which is translated by AUG, and then it'll end at a stop codon. Something really important you have to recognize is that the genetic code is redundant. And what this means is that a single amino acid may be coded for by more than one codon. So you'll notice that pro, here in the chart, is coded for by 4 sequences: CCU, CCC, CCA, and CCG. This is because the third base of a codon generally has some wiggle room to be different but not change the product. So this is why we call the genetic code redundant. And that is it for translation and transcription.